Okay, so we are carrying on looking at evolutionary epidemiology today. Last time we introduced this idea of evolutionary invasion analysis, and we used that to explore the evolution of drug resistance. Today is the penultimate lecture, so you guys are almost there in terms of, well, penultimate lecture in terms of new material. We've still got a quiz on the Friday and then a review lecture next week. Today we are going to be looking at virulence evolution. So we're going to be adapting some of the techniques or reusing some of them, learning a little bit more about the ones that we learned on Friday. And we're going to be doing this to try and understand virulence evolution, which is really a key topic within the evolutionary epidemiology literature. And virulence is all about trying to understand how the harm that a pathogen does to its host will evolve. Okay, so first of all, a little bit of a primer about virulence. So virulence is, so essentially you can think of the harm done to the host. So it could be just, how sick hosts get. It could be whether it causes mortality, death. So it could be the mortality rate or how quickly it causes mortality. It could be things as well, effects on host fecundity. So it's reproduction rate. Some pathogens can sterilize or reduce, sterilize the host or reduce their reproduction rate as well. So there are various different types of virulence. Today, we're going to be focusing on mortality virulence. This is the disease-associated death rate. We've not really been looking at models with disease-associated death rates in them because we've been usually trying to fix our population size, and so we don't have that varying. We can make some adjustments to account for these and kind of artificially keep our population size constant by accounting for these disease-associated deaths in our birth rate as well. So we can counteract that effect. You don't have to do that. Okay, so why might pathogens and more broadly parasites? I might occasionally flick back and forth between pathogens and parasites. That's because all pathogens are parasites in the broad ecological setting. If you remember all the way back when we introduced our first species interactions models, we talked about different types of interactions like antagonistic or mutualistic. Parasitism is a broad antagonistic lifestyle, if you like. And pathogens, we tend to think of microbes like bacteria and viruses, they're pathogens. But a lot of the stuff that we say about those pathogens also applies very broadly to parasites. So things like ticks and helminths that you wouldn't think of as being in this pathogen category. Okay. So we're going to be thinking about how the damage done to the host evolves. Yeah. And I don't want to cover anything else here. Yeah, maybe just back in the introduction slide quickly. I've mentioned this word here, phenotypes. Has anyone seen this word phenotype before? I know you will have, Scott, because we talked about it a lot. Anyone else? Is this familiar or not? Hands up who doesn't know what this is. Okay. So a phenotype you can really think about as being a trait or characteristic of an organism that it expresses. And you contrast this with something like a genotype. So a genotype is telling you something about the actual genetics, the genetic code, what nucleotides that a particular individual in a population has. It's genotype. Whereas a phenotype is telling you about what that actually relates to. So I've called this characteristics here. You can think of this as being traits as well. So, for example, it might be level of drug resistance is a phenotype. Or virulence, as we'll be doing in this lecture today. Also, things like, your eye color is a phenotype, your hair color is a phenotype. Okay. So what model are we going to be looking at today? Well, let's think about a pathogen that follows SIS type dynamics. So individuals are susceptible, they become infected, and then they recover, but then they become susceptible again. So there's no lasting immunity. That makes things a little bit simpler because we don't have to deal with a long-term immune class. We're going to assume that we have our density-dependent transmission, which we've been using a lot. Typically, we've been focusing on density-dependent transmission just because it's a little bit simpler to model than frequency-dependent transmission. It's also a little bit more broader. Most pathogens will fall into this category of density-dependent transmission. Frequency dependence just makes things a little bit more complicated. So that's why we've not been focusing on that. We've got our normal recovery rate gamma, and then now we're going to have this disease-associated death rate alpha. So we've not had this before. And we are going to include host demography into this. So host births and deaths. The reason being, if we didn't have any host demography, but we had hosts dying from disease, then we would eventually run out of hosts. Okay. So in order to keep this population size constant, what we're actually going to do is we're going to set our birth rate, B, equal to the death rate, D. We're dealing with proportions here as well. Remember, that's why there's no D times by N here. Well, to be honest, actually wouldn't matter at all. But it would factor into this I. We're going to have the birth rate equal to the standard death rate, which we've always had before by keeping our population size constant. And then we're just going to add on our disease-associated death rate times by the proportion that's infected. So this is basically just the disease-associated deaths, or total disease-associated deaths per unit time. And so this isn't individuals going from that I class back into the S class like recovery. This is just saying, okay, to keep our population size constant, we're going to roughly assume for every individual that dies, a new person is born. Okay, so what do our equations look like? Our dsdt is going to equal B, which is our birth rate, minus beta Si plus gamma I, so that's infections, recovery, and then natural mortality. Usually when I say natural mortality, I'm just saying non-disease associated mortality, or at least not this disease that we're interested in here. Then our di dt is going to be beta Si, it's infections, minus, and I'm going to put everything here in one bracket just to make it a little bit easier, disease-associated mortality alpha plus recovery gamma plus natural death rate D times by I. And this B here I've defined to be D plus alpha I. So these are births. Okay. So last time we talked about the concept of invasion fitness, and if we just take a quick recap on what we're doing here, we are starting off with this underlying population dynamics or ecological model, okay? What this is really saying is there's a particular pathogen with this transmission rate and this recovery rate and this mortality rate here. We're going to let that system sort of run for a while, and we're assuming that mutations are very rare, so that this reaches its endemic equilibrium, which happens to be stable. I'm not going to show that, but it is. So it reaches its endemic equilibrium, and then once it's reached that endemic equilibrium, we're going to introduce a rare mutant. And because that mutant is rare, this S here will become an S star. In other words, it's endemic equilibrium value. We just want to see if that rare mutant can invade or not. So that S won't be changing, and that allows us to simplify the dynamics a lot. What we're really doing here is we're testing the stability of that endemic equilibrium when we add in a new pathogen as a slightly different transmission rate, say, or mortality rate or recovery rate. And then we want to see whether that can invade. So it's essentially just a different way of doing stability analysis here. Okay, so that's the basic idea of what we're trying to do when we're looking at invasion fitness. Okay, so for this particular model, I'm not going to do these calculations. I'll leave this as an exercise if you want to do them, because we've done them quite a few times. Our basic reproduction number is beta over alpha plus gamma plus D. And this is just transmission rate in an entirely susceptible population. So number of new infections per infected individual when everyone else is susceptible, multiplied by the average infectious period. Here, the average infectious period is going to be 1 over the loss term, which is alpha plus gamma plus D. So that's our basic reproduction number. And just like the model we looked at previously, there is a stable endemic equilibrium at 1 over R naught, 1 minus 1 over R naught. Remember that our S and I here are proportions in this particular model. Okay, so we have those facts. That tells us something about this underlying ecological model here, ds dt and di dt. We're now going to be thinking about the invasion fitness. So what I want you guys to do is a bit of practice is to, if you need to, use your notes from the last lecture as a guide, write down an equation for the invasion fitness, which we call W. Before it was a function of gamma m and gamma, because we were looking at the recovery rate, because we were looking at drug resistance. Now we're going to be writing this as a function of alpha, so alpha m and alpha, where alpha m is a rare mutant with virulence alpha m, and alpha is the virulence of our resident pathogen. Okay, have a go at writing down that invasion fitness. And if you're not sure what to do, put up your hand and I'll help you. It needs to be based on this equation here. So that was for when we had recovery as the thing that's evolving. So you need to modify this equation now so that this equation here is frightened. Okay. Okay, let me go run through this with everyone. Okay, so what we're doing here is we're looking at our per capita growth rate. That's where this 1 over IM is going to be of our rare mutant. Our mutant we're calling, or the proportion of individuals infected with this rare mutant pathogen is slightly different in its level of virulence. We're going to be calling that IM. We want to look at our per capita growth rate. 1 over IM, DIM, DT. Really, we wouldn't usually write this in, but what we're doing is we're looking at this when S is equal to S star. You could even more properly write this as S star of alpha. Okay. Usually we wouldn't write this in just for sort of brevity, but just to make it clear what we're doing. We're evaluating this equation here when our S is fixed. And this level of S is determined by the resident level of virulence alpha. Okay. So what is this equation? Well, we just need to modify this equation up here. Wherever we see an I, we replace it with an IM because we're dealing with our mutant population, not our resident population. Our resident population is given by this I, not I sub M. And then because alpha is evolving, we replace alpha with alpha M. So we're going to have beta times by S star minus alpha M. We had this all in brackets. In fact, I'll take it out of brackets now. Minus alpha M minus gamma minus D. Check I've used the same notation. Yep. Okay, so that's my invasion fitness. This S star now is just 1 over R naught. So we can substitute that in. So what is 1 over R naught? 1 over R naught is going to be beta. It's beta here. Beta times by 1 over R naught is beta times by alpha plus gamma plus D. And this is the R naught for my resident pathogen. So that's why it's an alpha rather than an alpha M. And then we divide by beta because we're doing 1 over R naught. And then we've still got this minus alpha M minus gamma minus D. Then if you work that out, the betas cancel, the gammas cancel, and the Ds cancel, and you just end up with alpha minus alpha M. I have a show of hands who got that. Okay, a couple of people. Okay. So just to make sure it's clear what we're doing here when we're writing down invasion fitness. This is our baseline model. Whatever our baseline ecological population dynamics model is doing, we're saying, okay, we're assuming that reaches its endemic equilibrium or a stable equilibrium, and then we're going to introduce a rare mutant that differs in some trait or traits. Okay, so here we're taking this equation here, replicating it for a rare mutant, so it's almost the same, but differs slightly in one or more of these parameters. And instead of an I, in this case, it would be an I sub M because it's a mutant population. And then its fitness is going to be the rare growth rate. So we divide, it's rare per capita growth rate, so we divide by IM, have this S star in because we're at the endemic equilibrium, and then all I've done is that's just algebra. Okay. So the selection gradient. Remember, that was the first stage of what we did. We looked at the invasion fitness. That tells us if this value up here, this W, this function here, for a given value of alpha, the resident population, a given value of alpha M, so say this is a certain death rate of our current dominant pathogen in the population, a new pathogen comes along, a slightly different mutation, has a slightly different level of virulence. Can this one invade or not? If this is positive, then it's going to be able to invade because it's going to be able to grow when it's rare. And if it's negative, it's not going to be able to invade because it's just going to decay to 0. So what we want to do is now think about a process. That was just one particular case, right? We want about what's happening as a process of invasion and replacement. So remember I said before that if we think about these traits as just being sort of values along the line, this yellow line here, and what we're doing is we're saying, okay, we have a particular trait value, a particular value of virulence in this case, corresponds to where I'm stood. And we say, okay, we introduce a rare mutant, can it invade? Yes. Suppose it has a slightly higher level of virulence, it can invade and replaces it. And then we do exactly the same again, introduce a rare mutant, slightly different level of virulence, again, say slightly higher, can it invade? Yes. We keep doing that. We want to keep doing that until we get to a point where nothing else happens. Okay, and that's called a singular strategy. And so to look at that, we can look at something called the selection gradient. And that selection gradient is telling us which direction we're moving in. Okay, if my selection gradient is positive, then I'm going to be increasing my trait until I reach a singular strategy when there's that selection gradient is 0. On this side, if my selection gradient is negative, that trait is going to be decreasing. So that would be corresponding to over here. If it's increasing, then virulence is increasing. If it's decreasing, then virulence is decreasing. Okay. So looking at our selection gradient tells us which direction we're changing. Is it increasing or is it decreasing? So again, more practice for you guys. I want you to work out what is the selection gradient alpha. Selection gradient of alpha. Okay. So this selection gradient is just equal to our invasion fitness, W. We take the partial derivative of this with respect to our mutant trait. It's crucial that you do this with respect to the mutant trait and not the resident trait, okay? So taking the selection gradient is the derivative, partial derivative of W with respect to alpha M. And then after you've calculated the derivative, you set alpha M equal to alpha. And the reason we do this last stage here is we're assuming that there are very small mutations. So alpha M is approximately equal to alpha. And what we want to know is at a given point, are we increasing or decreasing? Okay. So if we do that for our fitness above, can anyone tell me what we're going to have? So this was our W that I've just highlighted, alpha minus alpha M. So if we take our partial derivative with respect to alpha M first, what are we going to have? Yeah, -1. And then in principle, yes, we would substitute in alpha M is equal to alpha. There aren't any alphas or alpha M's on this side anyway. And so our selection gradient, regardless of what our values of alpha and alpha M are, this is always equal to -1 and this is negative. So what does this mean? Like, how would you interpret this in terms of what's going to happen to the evolution of virulence in this model? Our selection gradient is always negative.
Left, regardless of whether it's greater or less.
Yeah, so what this means is that our mutant, so suppose we have a level of virulence here. Again, this is positive, this is sort of larger, and this is smaller, I should say. This is my current level of virulence. Mutants on this side that have higher virulence are not going to be able to invade. But if I'm here, mutants on this side have a lower level of virulence, can invade, and that is always true no matter where I'm stood on this line. Now, it doesn't make sense to have negative death rates. So what this means is that this will tend towards 0. So S of alpha is always negative. The selection gradient is negative for all alpha. So selection always favors lower virulence. In other words, alpha will tend towards 0 in the long term. So the question then is, in that case, why do pathogens harm their host? And you can think back to your R0 as well before we just move on. This should hopefully make some sense. Our R0 is essentially a proxy for this. If you make this alpha smaller and smaller, then you're going to be increasing R0, right? This is maximized when alpha is 0. Okay. That's because you're increasing the average infectious period, right? Killing the host doesn't do anything for the pathogen. So why do some pathogens, and most pathogens, harm their hosts? Well, the answer comes in because there must be usually some kind of trade-off. Okay. So we're going to be thinking about a trade-off between transmission and virulence. So in this model here that we've just looked at, only alpha was evolving. Now we're going to be thinking about what happens if there's a relationship between the virulence of a pathogen and its transmission rate. Okay, so why might that be the case? I go in through it here? No, I do not. So I'll talk about it here. So there's a certain degree of evidence that suggests that these kinds of relationships might exist. There's some counter evidence as well. But kind of mechanistically, it should make some sense that if a pathogen doesn't cause any damage to its host, that means it can't be infecting any cells, it can't be infecting any tissues, it can't be replicating. So anytime it's replicating, it has to be harvesting host resources or cells or tissues in order to produce, bacteria need to, produce more bacteria. To do that, they need to get their resources from somewhere. So typically they'll be infecting host cells and causing damage. If viruses infect a cell, typically they'll then burst and kill that cell. And that'll happen over and over and over again. And the more that happens, the more transmission stages that are produced, the more likely they're going to be transmitted onto the next host, but also the more damage done. So there should be some positive relationship between these things. So if we were to sketch an example of what this relationship might look like, if we think about our virulence going along this horizontal axis, and then our transmission rate being a function of that virulence, well, it could take different forms. Maybe it looks something like this. That would be saying that as our virulence gets higher and higher, our transmission rate might plateau, or it might just, not be accelerating as fast as our virulence is. We could see something like this, perhaps, where you get your transmission rate becomes higher and higher and higher, even if your virulence isn't becoming super high. Or you could see more complex things, maybe something like this. Okay. Or you have a bit of both of these things, depending on where you're at. There's a saturation effect, maybe. In all of these cases, though, what we want is this gradient, this function, to be positive at the very least. This curvature might vary, though. It wouldn't really make sense for a pathogen that was more transmissible to be less virulent, at least in this very simple model. There are actually scenarios where that does make sense. For example, if a pathogen infects the upper respiratory tract, so nasal passage and so on, tends to be less virulent, but also is more transmissible. So that's where sometimes this kind of relationship breaks down, but it can also apply in some cases. So that's what we're going to focus on. We're going to focus on the ones where it does apply. Okay, so let's go back to our model, right? We're using the same underlying ecological models. Whenever I'm saying ecological model, I mean this thing here, our basic population dynamics. This isn't changing, okay? This is just saying what happens if we've got these particular trait values. This is telling you the infection process, the birth-death process. That's not changing. What we're changing are these traits and the relationship between them. So that means that our underlying R0 is the same, and our underlying stable endemic equilibrium is the same. The only difference with that R0 now is that beta is now a function of alpha. Fundamentally, structurally, nothing else has changed. Okay, so what I want you guys to do now is to, again, write down an equation for invasion fitness. But now with this new relationship, beta is a function of alpha. Okay. So have a go at writing down your invasion fitness now. Okay, so it's fairly similar to start with. The main difference is that we've got beta of alpha m times by s star, rather than previously we just had beta times by s star. The reason this is an alpha m rather than an alpha is because this term here is telling us about new infections by our mutant. Okay, so new infections by our mutant have this transmission rate based on the mutant trait value. Okay, so those are new infections of our mutant, and then loss of that mutant by recovery or death is going to be minus alpha m plus gamma plus d. Just include it all in brackets there for now. And then we substitute in that s star, which is the same as before. So we'll have beta of alpha m times by alpha plus gamma plus d. The reason being that this s star is determined by our resident trait, which is alpha, not alpha m. And we're dividing by beta of alpha again. So the bit that I'll highlight here, this is 1 over R naught, and that's R naught based on our resident trait value alpha. So that's the first part. And then we have minus alpha m minus gamma minus d. And that's it. That's as far as we can reduce this. So previously we were able to reduce this a lot down to just alpha minus alpha m. This point here, this is as far as we can go. This is because of this trade-off here. So before we didn't have this trade-off here, and then those betas cancelled because they were the same thing. Now they're not the same thing. Because they cancelled before, we then lost the gammas and the d's as well. So we can see already that this trade-off has changed the invasion fitness, and therefore is going to change our selection gradient. So again, a bit more practice. I'll tell you what, no, I'll work through this one with you. I'll work through this one with you, and then I'll leave you the self-study problem. Okay, so let's calculate the selection gradient. So remember, this is the partial derivative of my invasion fitness, which I've just worked out in the previous slide, with respect to my mutant trait, and then evaluated when my mutant is equal to my resident. So what's this going to be? Well, we don't know what this function beta is. It's just some function of alpha. So what I'm going to do is I'm going to do this in 2 stages in the same place. So you might want to hold off copying this down. I'll write down this unevaluated first of all, and then evaluate it. Make sure it's clear. Okay, so unevaluated, we take the derivative of this function up here with respect to alpha m. Well, the bit that's highlighted is just a constant, right? Because this is a function of alpha, not alpha m. So all we're going to have here is beta prime of alpha m. And I'll leave it just as s star for now, just because it's a little bit easier to write it down. So beta prime of alpha m times by s star. And then these terms here, well, the gamma and d, they're not functions of alpha m, so they're just going to drop out, right? And we've got a minus alpha m here. So I'll have a -1 as the derivative. That. And then I said I'd evaluate this now when alpha m is equal to alpha. So everywhere I'd see an alpha m, I just replace with an alpha. The only place is just here. So this here goes beta prime of alpha. So beta prime of alpha, so the derivative of our trade-off times by our endemic equilibrium number of susceptibles or proportion of susceptibles, S star, then -1. This is my selection gradient. We could also write this as beta prime of alpha. S star was 1 over R naught. Because this S star is a function of alpha itself, I'm going to write this as 1 over R naught of alpha, just to remind us that this R naught is a function of alpha. Okay. And then -1. So remember, this selection gradient is telling us the direction in which we are evolving. So if this value here of S of alpha is positive, then alpha is going to be increasing. And if it's negative, alpha is going to be decreasing. And if it's equal to 0, then we're at a singular strategy. We refer to this as alpha star. So what must our singular strategy satisfy, regardless of whatever this function is? We just go back here for a second. We've not stated what this relationship is between beta and alpha, right? We've said it could be anything. But at a singular strategy, it must satisfy this being equal to 0, regardless of what that trade-off is. So at a singular strategy, our selection gradient at that singular strategy must equal 0. By definition, that's what it means for it to be a singular strategy. It's saying that at that point, selection is neither pushing it to increase or decrease. So you can think of this as being like an evolutionary equilibrium. So we've got an equation for S of alpha above. So you know that beta prime of alpha star over R naught of alpha star -1 must equal 0. In other words, beta prime of alpha star must equal R naught of alpha star. Now this is general for this particular model, regardless of what our beta prime is, what our relationship is. This is telling us that the gradient of my trade-off must equal R naught to be satisfied. This is not a sort of universal general finding across virulence evolution models. This is for this particular model that we've been looking at, but it works for any trade-off. Okay, what I thought I'd leave for today is the self-study problem. I'd leave the last 10 minutes for you guys to work through this. And again, just ask me any questions if you have them. So yeah, have a go. First of all, what we're going to do is we're going to pick a particular trade-off. We're going to say, okay, we're assuming that the transmission rate is equal to the virulence, the mortality rate, square rooted. Okay, so we've got a particular function for beta now. That means we can work out what beta of alpha is. We can work out what beta prime of alpha is, rather than just using these general functional forms that we had up here. So we can substitute those in. First, I want you to have a go at sketching this trade-off, just to check you can see what type of trade-off it is, what's it doing, and then find the singular strategy alpha star. So find an explicit value for alpha star. And then if you have time, or in your own time at home, have a go at sketching the selection gradient itself and deduce that the pathogen will evolve to alpha star. So this is a little bit more complicated, but think about that idea of if your selection gradient is positive, then alpha will be increasing, and if your selection gradient is negative, it will be decreasing. Okay, have a go at those few things. I'll walk around and answer any questions you guys have. I will leave, I'll zoom out a bit. I will leave there we go. So there's the selection gradient up there at the top, and then this is the selection gradient evaluated at the singular strategy. This first part here is not a trick question, by the way, the sketching the trade-off. It's literally have a quick, remember what the square root function looks like. That's all that is. There's nothing too complicated here. The more tricky part that I want you to spend more time focusing on is finding the singular strategy, okay? The first part's really just to remember what sketching a graph looks like. Okay, I'll work through this second part here, just because I want to make sure everyone's kind of clear on how we find the singular strategy in this scenario where we've got an actual trade-off rather than just something generic. Okay, so I'll work through the second part. So we've got beta of alpha is equal to square root of alpha, which is just alpha to the power of a half. So if we take the derivative of this, half comes to the front, lower the power by 1. So we just have 1 over 2 root alpha. Okay. So that's the derivative of my trade-off. Why do we want that? Well, that features in our selection gradient. So S of alpha, which we worked out on the previous slide, was beta prime of alpha over R naught of alpha minus 1. And I defined R naught of alpha to be beta of alpha over alpha plus gamma plus D. Okay, so let's substitute those things in. So beta prime of alpha is 1 over 2 root alpha. We're multiplying that by 1 over R naught alpha. So 1 over R naught alpha, we're going to have alpha plus gamma plus D in the numerator, and beta of alpha on the denominator. And beta of alpha is just root alpha. And then we've got this minus 1 at the end. So 2 root alpha times by root alpha is just going to be 2 alpha. So we're going to have alpha plus gamma plus D over 2 alpha minus 1. So that's my selection gradient. And then if I want to find my singular strategy, I need to set this selection gradient equal to 0. I'm calling alpha star my singular strategy that satisfies this equation. In other words, alpha star plus gamma plus D over 2 alpha star minus 1 must equal 0. If I multiply throughout by 2 alpha star, I'll get alpha star plus gamma plus D minus 2 alpha star is equal to 0. Or gamma plus D minus alpha star is equal to 0. In other words, alpha star, my singular strategy is equal to gamma plus D. This is telling me that my optimal level of virulence is going to be equal to my recovery rate plus my actual mortality rate. This is all ultimately about average infectious periods. You remember your average infectious period is alpha plus gamma plus D. This is basically saying the component that comes from disease-associated mortality has to be equal to the other loss terms. This also tells you something potentially profound. If you increase the recovery rate, you should increase selection for virulence. So if individuals are recovering faster, then you'll increase selection for virulence. So potentially doing things like providing more treatments could increase selection for it to become more deadly. Also, if you have a higher population turnover rate, if you have a higher death rate, that will also increase selection for virulence. So this kind of equation here, okay, yeah, there's a mathematical equation. It also tells you something about the underlying biological relationships. Okay, that's it for today. I would encourage you still to do that, the final part of that self-study problem, and thinking about the selection gradient and how that's drawing a sketch of that, drawing an accurate sketch of that tells you something about the direction which you're evolving. Okay. Any other questions? I'll stick around for a few moments. If not, I'll see you guys on Wednesday for the final lecture of new material. Okay. Have a good week.